In this series of videos, I'll describe the steps you would take if you wanted to express a mammalian gene, which I will call your favorite gene, in the bacterium Escherichia coli. Expressing a gene in an organism means that the organism makes many messenger RNA copies of the gene of interest, and then the organism translates that mRNA to produce the protein product of the gene. The major steps in the process are shown in this diagram. The process of making many copies of a gene in a microorganism is often called cloning the gene. As I describe this process, I'll introduce you to many concepts and techniques in recombinant DNA technology. My goal is not to describe these technologies comprehensively, and many technologies exist that I will not even talk about. But this series of videos will provide a fundamental understanding of many basic technologies used to manipulate and analyze DNA. The significance of these technologies is enormous, as they allow us to engineer living organisms to have desirable properties or abilities. These technologies have moved us into the era of synthetic biology. Some of the techniques I will talk about rely on denaturation and hybridization of DNA, so I want to spend the rest of the video going over these processes. As you know, DNA consists of two complementary nucleic acid strands that are held together by non-covalent interactions between the nitrogenous bases in the strands. When you heat double-stranded DNA to a high enough temperature, the interactions between the strands are broken, and the strands separate from each other. This process is called denaturation. You can follow this process by monitoring the ability of the DNA solution to absorb light at a wavelength of 260 nanometers. Single-stranded DNA absorbs light more strongly at this wavelength than double-stranded DNA, and so as the DNA denatures, its absorbance will increase. You can see that this change in absorbance is not instantaneous, but will occur over a range of temperatures. The temperature at which DNA is half double-stranded, or half native, and half single-stranded, or half denatured, is called the melting temperature, or Tm. This process is reversible, such that if you cool the DNA, the two strands can anneal to each other, or renature. But you have to be careful, because if you cool the single-stranded DNA too quickly, the bases in one strand may not be able to find their proper base pairing partners in the other strand. This would result in a jumbled mess of improperly annealed DNA. So, if you want to obtain proper base pairing, it's better to cool the DNA slowly or to a temperature just a bit below the melting temperature at first. This will allow improperly base paired strands to separate from each other now and then, giving them a chance to find their correct base pairing partners. For various reasons, we often want to anneal small fragments of DNA to each other or to longer pieces of single-stranded DNA. DNA synthesis machines have been invented that allow us to easily make oligonucleotides several dozens of bases in length. So it's important to realize that we can denature long sections of double-stranded DNA and then cool them in the presence of shorter synthetic oligonucleotides. Some of the short oligonucleotides will anneal to the longer single-stranded DNA. This process also works for DNA and RNA. If you synthesize some DNA that is complementary to an RNA molecule of interest, you can create double-stranded DNA-RNA hybrids. The major factors that influence the temperature at which an oligonucleotide will anneal to a longer nucleic acid are listed here. You should understand the influence each factor has on melting temperature. All other things being equal, a longer oligonucleotide will melt at a higher temperature than a shorter oligonucleotide. GC-rich regions of DNA bind to each other more tightly than sections containing a lot of AT base pairs. Including a mismatch in the oligonucleotide, such that the two strands are not perfectly complementary, will lower the melting temperature. A high concentration of salt in the aqueous solution in which the DNA is dissolved will increase melting temperature. Note that the hydrophobic surfaces of the nitrogenous bases are mostly hidden from the environment in the double-stranded form of DNA. A high salt concentration increases the polarity of the environment, making it less favorable for the hydrophobic surfaces to be exposed through melting of the DNA. Extremes of pH will change the protonation state of the bases, hindering their ability to base pair. so DNA is most stable near the middle of the pH scale. And finally, organic solvent in the environment decreases the polarity of the environment, making it easier for the DNA to denature. Therefore, DNA is most stable if no organic solvents are present. In the next video, I'll start describing the process of cloning your favorite gene.